Hello, I'm Dr. Maryam Nesvaderani from the University of Sydney Nepean Clinical School in Australia, and I'm going to talk to you for a few minutes about a project we've done together with the University of British Columbia Department of Immunology and Microbiology. So this is a study about severe pancreatitis, and we were trying to find a biomarker that could predict severe pancreatitis within the first 24 hours of admission to hospital. This is important because severe pancreatitis has a high mortality rate, and treatment decisions undertaken within this early time frame of 24 hours can improve patient outcomes. For example, starting enteral feeds early or admitting the patient to an intensive care unit. Currently, there's no system that reliably predicts severe pancreatitis in this early time frame. The scoring systems that we have usually take about 48 hours to provide an accurate evaluation. So for this study, we had 87 patients. 10 of these had severe pancreatitis, 20 had moderately severe, and the rest had mild. And we had some control subjects too. So we collected blood from these patients on presentation to the emergency department and performed an RNA sequencing study. So RNA sequencing is a next generation transcriptome sequencing technique, and it gives us a snapshot of the RNA expressed within those cells at a particular point in time. So we were looking at the peripheral blood cells of these patients with pancreatitis in that early 24-hour time frame on presentation to hospital. And our aim was to find transcripts that were unique to severe pancreatitis that may be used as a biomarker in the future to help predict severity on admission to hospital. What we found was interesting. So there were 422 differentially expressed genes in the severe pancreatitis group, and the number of differentially expressed genes increased as severity increased. There were actually no differences in gene expression when we looked at the peripheral blood cells based on etiology. So there was no difference in these cells between gallstone, alcohol, or idiopathic pancreatitis. This suggested that there's some final common pathway that occurs in pancreatitis independent of the initiating cause. We then use pathway analysis to look for molecular mechanisms that were present in the severe group that weren't there in the other groups. And what we found was that there was some immune-related pathways unique to severe pancreatitis, and these were related to neutrophil function as well as type 1 interferon signaling. We also identified a 4-gene signature or endotype in this study, and these 4 genes could classify patients with severe pancreatitis with an accuracy of 64% in this 24-hour early period. This is an important first step in being able to predict severity within this early stage. This study has also shed light on the molecular pathways behind severe pancreatitis, and this will be useful in the future for designing targeted therapies for pancreatitis, because at the moment we mostly rely on supportive treatment. We've identified a relationship between severe pancreatitis and sepsis. So those 4 genes that gave a signature for severe pancreatitis all have links to sepsis. And our findings support the use of immune modulating therapy in the future as a targeted therapy for pancreatitis. Thank you so much for watching this short video summarizing our paper. Have a look at our paper if you'd like to see all the fine details about this study. Thank you.